Integration in logical learning.
Flowering plants, Class 9 ICSE. Vegetative propagation. Vegetative propagation is a form of asexual reproduction of a plant. Only one plant is involved and the offspring is the result of one parent. The new plant is genetically identical to the parent. Types of vegetative propagation are natural vegetative propagation and artificial vegetative propagation. Now, what is natural vegetative propagation? New plants grow from parts like stems, roots, leaves of the parent plant. Stems. Runners are the stems that grow horizontally above the ground. They have nodes where the buds are formed. These buds grow into a new plant. Vegetative plant structures that arise from the plant stems include rhizomes, runners, bulbs, tubers, and corms. Roots. New plants will grow out of swollen, modified roots called tubers. Buds develop at the base of the stem and then grow into new plants. Leaves. Leaves of some plants will grow into a new plant if they become detached from the parent plant. Other plants grow small plants called plantlets on the edge of their leaves. In Bryophyllum, plantlets develop from the buds present on the marginal notches of the intact leaves. These plantlets become detached and develop into independent plants. Artificial vegetative reproduction. Horticulturists and the farmers use artificial means to produce the plants that are identical to the parent plant. Some of the methods used are cuttings, grafting, and layering as well. Now, what are cuttings? Cuttings are a part of the plant that is cut off of the parent plant. Shoots with leaves attached are usually used. New roots and leaves will grow from the cutting. The shoot is cut at an angle. A growth promoter may be used to help with the growth of the roots. Grafting. In grafting, two plants are used to develop a new plant with the combined trait from the two parent plants. In grafting, the scion is the above ground part of one plant. The scion is attached to the stalk, which is the rooted part of the second plant. The layering process is shown here below. Layering. In layering, a shoot of a parent plant is bent until it can be covered by the soil. The tip of the shoot remains above the ground, new roots and eventually a new plant will grow. These plants then can be separated. Advantages and disadvantages of vegetative propagation. Advantages of vegetative propagation are they are quicker and more certain, produces identical quality as the parent. Plants that do not have a viable seed can be reproduced. Flowers produced are of superior quality. Desirable character of fruit can be maintained. Now, what are the disadvantages? It does not produce new variety. It leads to overcrowding around the parent plant. Very little possibility of dispersal. Economic importance of the artificial propagation. Artificial vegetative propagation is the process of growing plants applying artificial techniques such as layering, grafting, and cutting. This method is more beneficial as it boosts the quality, yield, and the disease resistance in plants and thereby the plant products. Thus, this method is economically viable. Hybridization, when the two different varieties of the species of plants are mixed to get a new breed of plants containing the characteristics of both the parents, the process is called hybridization or crossbreeding. Hybridization can be intervarietal, interspecific, or intergeneric. It can be done by cross-pollination or somatic cell hybridization. Now, what is somatic cell hybridization? I think we all know that the vegetative cells of two different plants, isolated, cell walls are carefully dissolved and the cell contents are fused together, and then the fused cells grown under ideal conditions replanted when they develop into small plants. Example, triticale is developed from the tritium or wheat or the rye or the sea kale. Benefits of hybridization. Hybrid plants are generally disease resistant and pest resistant, can grow on different types of soil, show early maturity and produce larger yields, more nutritional and economic value, and shorter life cycle. Micropropagation. Micropropagation is the aseptic culture of cells, pieces of tissue or organs. It is possible to regenerate new plants from small pieces of plant tissue because each cell of a given plant has the same genetic makeup and is totipotent and that is capable of developing along a programmed pathway leading to the formation of the entire plant that is identical to the plant from which it was derived. The process of micropropagation can be divided into four stages. Initial stage, a piece of plant tissue called an explant is cut from the plant. Disinfested, removal of surface contaminants and then placed on a medium. Now the multiplication stage. A growing explant can be induced to produce the vegetative shoots by including a cytokinin in the medium. A cytokinin is a plant growth regulator that promotes shoot formation from growing plant cells. Rooting or pre-plant stage. Growing shoots can be induced to produce adventitious roots by including an auxin in the medium. Auxins are the plant growth regulators that promote the root formation. For easily rooted plants, an auxin is usually not necessary and many commercial labs will skip this step. Acclimatization. A growing rooted shoot can be removed from the tissue culture and placed in the soil. When this is done, the humidity must be gradually reduced over time. Because the tissue cultured plants are extremely susceptible to wilting. Uses. Plant with long seed dormancy or slow breeders are grown this way. Disease-free cells can be isolated and used to yield disease-free plants. Offspring genetically similar to the parents. Application of biotechnology. Biotechnology is a stream in biology which deals with the application of technology to biological processes occurring in the living entities or their subordinates to transform a particular process for their specific utilization. Insulin production. Insulin is a hormone that regulates the amount of glucose or sugar in the blood and is required for the body to function normally. Insulin is produced by the cells in the pancreas called the islets of Langerhans. Without the insulin, the blood glucose builds up in the blood and the cells are starved of their energy source. People who do not produce the necessary amount of insulin have diabetes. People who do not produce the necessary amount of insulin have diabetes. So, in the same way, if the body does not produce any or enough insulin, people need to take a manufactured version of it. The major use of producing insulin is for the diabetics who does not make enough or any insulin naturally. Synthesizing human insulin is a multi-step biochemical process that depends on basic recombinant DNA techniques and an understanding of the insulin gene. DNA carries the instructions for how the body works, and one small segment of the DNA, the insulin gene code for the protein insulin. Manufacturers manipulate the biological precursor to insulin so that it grows inside the simple bacteria. Penicillin production. Penicillin is a secondary metabolite of the certain species of the penicillium and is produced when growth of the fungus is inhibited by the stress. It is not produced during active growth. Penicillins are the antibiotics used to treat the bacterial infections that are derived from the antibiotic penicillin. Penicillin fermentation is an aerobic process. The required aeration rate varies according to the strain, the type of the fermenter used, and on the impeller system. It is produced by fed batch submerged fermentation in a stirred tank fermenter. Tetracycline production. Tetracycline is a broad-spectrum polypeptide antibiotic produced by the Streptomyces genus of the Actinobacteria. Tetracycline antibiotics are the protein synthesis inhibitors. Tetracycline is an antibiotic used to treat many different bacterial infections such as urinary tract infections, acne, gonorrhea, chlamydia, and others. Alcohol production. The production of alcohol beverages is a process that involves the active participation of microorganisms, most often yeasts. Yeasts are the main fermenter and the alcohol producer in the production of wine, beer, and other alcohol drinks. The main yeast species used is Saccharomyces cerevisiae. It ferments the sugars coming from the different sources, example grapes for wine, barley for beer, to alcohol and carbon dioxide. Both wild and cultivated strains are used. The species or the strains used in the fermentation play an important role in giving the final taste properties of the drink. Vinegar production. Vinegar is a food product made by the acetic acid bacteria that can ferment the alcohol in an alcoholic liquid to acetic acid. The fermentation of alcohol requires oxygen and its availability can determine the rate of vinegar production. The main genus used for vinegar fermentation is Acetobacter sp. Since the final product of its fermentation can contain acetic acid as high as 20%, some of the most common uses of vinegar are in food preparation as a cleaning agent and as a medicine. Now the yogurt production. Yogurt is the result of the fermentation of the two main bacterial species, Lactobacillus bulgarisus, Streptococcus thermophilus. Sometimes the other lactic acid bacteria are added as well. Yogurt is most often made of the cow's milk, although milk from sheep, goat, water, buffalo, camels and yaks are used as well depending on the region of the cultivation. Now let's talk about the cheese production. Cheese is another popular and ancient dairy product. It consists of milk, proteins and fat together with lactic acid bacteria. It has longer shelf life than the uncultured milk. Currently, there are few hundred varieties of cheese produced all over the world. Making cheese is similar to yogurt, but after the acidification, usually with the lactic acid bacteria. Saccharomyces cerevisiae produced and sold as baker's yeast, although some artisan bakers maintain their own starter cultures. Vitamin C production. Vitamin C or the ascorbic acid plays an important role as an antioxidant and in collagen synthesis. It is produced from the glucose by a combination of chemical and biotechnological steps. Now the last, lipase enzyme production. What is this? Enzyme production fermentation technology could be a field that involves the utilization of microorganisms and enzymes for the production of compounds that have application within the energy, material, pharmaceutical, chemical and also the food business. Lipases are enzymes which catalyze the hydrolysis of long-chain triglycerides. They are naturally present in the stomach and pancreas of humans and other animal species in order to digest fats and lipids. Microbial lipases are produced by the bacteria, fungi and yeast. Thank you for watching this video.